Hey everyone, this is James Roguski, and today is August 13th, and I'm here with Roger Meacock. And the purpose of this is to try to capture lightning in a bottle twice. Roger gave a presentation that I was blessed to be able to witness, and I asked him to come on so I could give him access to all the folks that read my work, because I think that everyone needs to know what Roger's going to share. And so, Roger, the floor is yours. Please share your wisdom and your work with everyone that may get a chance to see this.
Thanks, James. Just quick introduction to me. I'm a holistic vet here in the UK. I became aware of the potential dangers of the mod RNA vaccines. I refuse to call them mRNA because that's a natural thing that occurs in the body, and they've been made with pseudouridine, so they're not the same. Um, so I became aware of the potential dangers of those, and I reached out to somebody that I'd been following through the COVID um, sort of online discussions, which was Dr. Kevin McCairn, who's a neurosystems a scientist, and asked him if the potential was the same danger for animals as humans, and he basically said yes. So we put together a letter, an open letter of concern to the Veterinary Medicines Directive, who oversee medicines in the UK here, and put them on notice effectively in July 2023. So this is a little presentation of the potential dangers in veterinary medicine if mod RNA jabs were to be used in animals. So away we go. Right. So we have, that's me, that's mine practice, Natural Healing Solutions. That's what I call myself. And this was a presentation, as James said, that I did for the World Council for Veterinary Health that I was talking to for the Canadian branch of the World Council for Health to try and promote the veterinary side of it, which we're looking to start, and the dangers of mod RNA and prions. So the mod RNA jabs are inherently dangerous. As everybody knows, I'm sure already, they are not vaccines. They are gene therapies, which was included in the BioNTech SEC filing. So that's their own technology definition, not mine. The lipid nanoparticles can cause inflammation, cause emboli, which is clotting through clumping and allergic reactions. And the LNP mod RNA does not stay in the injection site, as we were told. We know now that it goes all around the body, especially to the gonads. So that's the ovaries and the testes, lymphatic tissues, and the brain and nerves, etc. So mod RNA can enter the nucleus and can reverse into the genome. And due to the pseudouridine, the body doesn't stop protein manufacture. So mod RNA will be broken down and may mimic microRNA cell signaling molecules, which are something that we've only really relatively recently become aware of in terms of their role in regulation of the biology of the body. And they're very short strands of RNA. So as you can imagine, if you've got a longer mod RNA molecule, as it's broken down, some of those smaller strands might suddenly become active and have roles interfering with cell signaling. And we know in the human body, there's about 5,000 of these microRNA that have been identified, of which we probably only know in detail less than a handful, less than 10. And so to know how each of those small microRNA strands that might be produced from the mod RNA breaking down, what effect they might have in the body and interfere with that delicate branch of a network between them all. We've got no way of knowing that. I'm not even sure if we had a super quantum computer, it would be able to calculate all the interactions of 5,000 different microRNAs, and that was if you just interfered with one. But if breaking down a mod RNA could potentially produce more than one microRNA that could have all sorts of interactions. So it's really messing with biology in a way that we don't yet fully understand. and it's an arrogance to think that we do. There is also a prionogenic risk of mod RNA itself and whatever protein it directs the body to make. Prions, I will come and explain those in a little bit in a while, but they're obviously, if people have heard of mad cow disease or Creutzfeldt-Jakob disease in people or scrapie in sheep, that's the sort of diseases that we're talking about here. There's also a risk potential of autoimmunity if the protein structure mimics any of the proteins naturally occurring in the body. So if we talk first about the mod RNA in the food livestock. So there is currently one mod RNA product in the USA already for pigs called Sequivity. It was somehow licensed in 2018 before the definition of a vaccine had been altered. So I'm not quite sure how that one was got through past FDA to get its license, but that's what happened. And it's a product platform, not a specific end product. So if, for example, there's an outbreak of respiratory disease on a pig farm, a vet will go along and they'll swab the various different pigs, send the swabs off to a laboratory. The laboratory will identify the pathogen and sequence it, and then choose a gene and then a protein of interest that they will then try and get the mod RNA to generate within the body. So they'll make these mod RNA vaccines, jabs, to send back to the farm, and then the farmer will inject them into the pigs. Now, that whole process from start to finish, from swabbing back to the point where the farmer's injecting the pigs, is 8 to 12 weeks, which, as you can imagine, is nowhere near long enough to do any specific safety checks and make sure everything's okay. And there's also a declared intention to move animal vaccination to the mod RNA jabs as well. So it's it is something that's definitely in the pipeline if we don't manage to cut it off at the pass. So they have an inherent risks that aren't being checked. As I say, 8 to 12 weeks isn't long enough by any means. So if the chosen protein or the selected on mod RNA itself is prionogenic, i.e. it will cause the formation of these prions that cause these prion diseases, then it will initiate the protein misfolding and the prion formation. And that's what happens with prion diseases. You get a PRP protein which naturally occurs in the body, and it starts to misfold. And it's a little bit like a crystal in a, if you were trying to grow a crystal at school and in chemistry lab, you put a seed crystal in a strong solution, and over time it settles out and the crystal grows and grows. And that's effectively what happens with these prions within the body. Also, we could potentially get prion shedding that could affect the microbiome and then into the environment. And if an infected animal is slaughtered before symptoms manifest, the prions will be in the food chain and could potentially make meat for food infected for humans and also our pet carnivore cats and dogs. So, given that the lifespan of say pigs and poultry especially is so short, they wouldn't necessarily have time to manifest any prion diseases themselves that we would then see symptoms of to know that there was a problem. So the first time we might know that there is an issue might be when people actually started getting these prion diseases, and then somehow they would sort of work back and work out where they'd come from. So the mod RNA and prion hazards. So the mod RNA itself can be prionogenic. The chosen protein produced under instruction may be prionogenic, but we also know that there's a frame shifting errors that can happen due to the pseudouridine that is used within the mod RNA projects, which produce unintended proteins which may be prionogenic. So you can also have functional epitopes that have unintended consequences, which make them all the more dangerous. So even if the protein was checked out, if something were to be looked at in one way, but the protein sort of started to break down, it might reveal another site that might be prionogenic as well. So the proteins may shed in the milk, sweat, urine, feces, saliva, potentially to the environment. and the prions may shed too. Prions in the slurry used as fertilizer go to the soil and the soil fauna. Plant roots can uptake prions from the soil and accumulate in the leaves and stems. So the prions in the soil will leach down into the water system, and they're not neutralized by water processing and could potentially spread like microplastics do in rain. I mean, who knows? We don't really know. Obviously, if people are worried or thinking that they're safe because they're vegetarian or vegan and they don't eat meat and therefore they can't get prions from the meat, the fact that the prions can go to the soil and then into the soil fauna and then potentially even into the leaves and stems, means that even plant food could potentially be a problem. So this is just a pictorial demonstration of what I've seen. So we've got a mod RNA there, which produces the protein. So this is the PRP protein in the body, which can be changed into the scrapie form either by the mod RNA or the protein itself. And from this folded protein can then potentially go into the topsoil, and then from there it could go to the water, or it could go to the roots of the plants and then go to vegetables, or into grass, so for grazing. So you could get chronic wasting disease, which happens in deer, or potentially BSE in cows or scrapie in sheep. So this is actually one way that chronic wasting disease has already been found to spread through these grazing idea. So it's not just theoretical, it's actually been shown to happen. So the potential consumer dangers. So the consumption of prion infected meat from farmed or wild animals. So those of you who go hunting, if you're shooting deer, be careful of what this chronic wasting disease in your where you're hunting is. Make sure that there isn't any there, or it's not close, it can't possibly spread. Obviously, consumption of prions in the fertilized crops affected by slurry or water. Slurry is like it's like the manure from the cows if it's used on crops. Consumption of prion infected water and dairy produce. Consumption of prions that have passed through other species can also accelerate misfolding activity. So, for example, if a deer were to eat the BSE prion, which picked up from crops, and then the deer was eaten by humans, then the human would actually get a more rapid and more severe form of the BSE because of the fact that it passed through the deer. Why that happens, I don't think anybody knows, but it does. So given that the number of animals that could potentially be being involved with a number of vaccinations that were being given, because of risk of new and previously unknown prions, and even the development of something called a super prion, which has no species specificity, we just don't know what might be developed. So there's previously unconsidered risks from surgical instruments. So even if vets are operating on animals, they could potentially move them, from one to the other, or even from farm to farm, and also claving isn't sufficient to get rid of the prions. Blood transfusions and organ donation and exosome shedding, obviously, these are sorts of things that could happen in a small animal environment, but it's nonetheless a risk. There's also the potential for prions to interfere with P53, which is the body's natural defense for cancer. So obviously, if P53 is interfered with, then our cancer protection drops, and then we'd see a higher rate of cancer. So the difference compared with what we saw in the UK with BSE in the 1990s, which I had the unfortunate honour of seeing cows with that when I was not long qualified. So, with whole herd mod RNA inoculation, it increases the chances of prion formation. Highly indestructible nature of prions will tend to cause concentration accumulation in the environment and the animals. and the inability and impracticality to filter prions or to neutralize them with heat, etc., will lead to increased speed of accumulation and concentration. The sheer numbers of potential animals involved in the event of a mod RNA jab is mandated. So the World Health Organization have what they call a One Health policy. And I know James will have mentioned this in his talks about the International Health Regulation amendments and also the pandemic treaty intended. And part of that could potentially be to mandate some sort of veterinary intervention in the animals. So obviously, if that was mandated worldwide, the risk just multiplies up. And given the people's reluctance now to take the mod RNA jabs and existing government contracts, animals could be seen as a contractual solution to save the government from having to default on their contract, and pushed out under the excuse of potentially even countering antimicrobial resistance. And there has been a recent paper, I think, from one of the Israeli universities where they've actually spoken about using mod RNA for bacterial infections to avoid antimicrobial resistance. So, although I wrote this before that paper was published, I was predicting the potential of it, and it sort of happened. So if we come on to the mod RNA for avian influenza, bird flu, there are currently trials underway for the mod RNA jabs for avian influenza in the USA, including a self-amplifying RNA. So that means that the body actually produces the RNA in an uncontrolled way, and then those uncontrolled RNAs then produce protein in an uncontrolled way. So we've got two stages of uncontrolled amplification. So, what could possibly go wrong? So, self-amplifying. So we produce, already done that explanation. So RFK Jr.'s halt to mod RNA technology has stopped some, but not all trials and developments. So certainly the self-amplifying trials are still going ahead. And there's also trans-amplifying RNA, which is being developed and researched as well. So, it hasn't been properly stopped. Once we've understood that this platform is unsafe, that there should really be no exceptions to halting it. And the same risks here apply as for sequivity, where flocks of birds could be swabbed and then jabs sent out in a very short space of time. And of course, when you've got different products being sent out under the same name, it makes pharmacovigilance almost impossible, because you're not comparing like with like in terms of the results. So it's it's just everything is set up to let these prion risks go, let that slip through the net. If we're coming on to pets, because obviously a lot of people are worried about what could potentially happen to their pets. If you can hear my puppy squeaking in the background, I apologize. He's just 12 weeks old. So there are already mod RNA jabs being used in North America primarily. So we've got rabies in Canada, canine influenza in the USA, Mexico, Chile, Philippines, and from June 2024. And there's the cat feline leukemia virus in the USA from February this year. Not in the UK yet, from anybody who's watching from the UK. So these are made by Merck MSD, and they're now calling them RNA particle vaccines to try and sort of hide what they are. And obviously people are very much aware now of the mRNA or mod RNA vaccine. So by calling them something slightly different, it's designed to confuse. And they're launched already under the brand name Nobivac NXT vaccines. Now, Nobivac is a brand name that's already been used for traditional vaccines for years. So, they're really trying to slip these self-amplifying vaccines in under the radar by calling them something slightly differently, and also then reversing them into an existing brand. So given that shedding has been proven to occur from the mod RNA COVID jabs, we must consider that it's a risk for these. as well, so potentially to other pets and potentially owners as well, and we don't know what effects that might have. So as I say, the NXT are self-amplifying RNA jabs. So the consequence of prions. So firstly, research shows there is no safe level of prion. It's just a question of dose. Dose determines the speed at which it becomes a problem, but there is no safe level. So prions and amyloid. Amyloid is another protein that can occur in the body and misfold through a very similar process. So you get a renal or cardiac amyloidosis, where different organs get effectively furred up with this protein misfolding. And enzymes can break it down, but the breakdown products can retain prionogenic activity as well. So there are potential treatments. Then the implication for this is potential treatments need proper testing in case they help spread and seed the prion activity around the body via exosomes and immune cells. So if people are taking proteases to try and break these down, just be careful that if you've got one sort of seed crystal, if you like, if you break it up, you'll actually end up potentially have 4, 5, 6, 10, 20, however many seed proteins in this case that will set off the misfolding in other areas too. And this, people are recommending these in all good faith, but we just need to be careful what we're recommending and check them out properly before we sort of dive in with them. I'm a great believer, if it ain't broke, don't fix it. So if you haven't got any symptoms, don't necessarily feel that you have to detox from spike protein or whatever else. Just do what you need to do as you need to do it. So as I said before, prions are resistant to heat and other preventative practices, and they can't even be really sterilized. There have been cases of humans getting transmission via surgical instruments. So the delay in disease onset means treatments often start too late, and what treatment works for one may not work for all, because there may well be, with different prions and different proteins potentially being made by all these mod RNA jabs, they won't necessarily all behave the same. We've got new variant CJD, but there could be however many variations of new variant CJD. There could be 100 variations of new variant CJD. We just don't know. It's an unknown field. But just so that people might think that I'm scaremongering and I've got no basis for saying what I'm saying, well, this is a graph that was produced by DataBridge, who do market research and market predictions for medicines for the pharmaceutical industry, so that they know what to invest in for the future. And we can see here that they're envisaging there's going to be quite a rise in prion diseases, certainly by 2030. So, there must be a reason why they think that prion diseases are going to be increasing in this way, and that therefore pharmaceutical companies should be investing in them to do what they do. So, it's not me that's scaremongering here. This is actually information coming from the pharmaceutical industry itself that's sort of backing up what I'm saying. So part of what I was talking about on Friday, see, my concern is for the animals and the health, for their health, and also for our health as well. And with this end, I've been asked to help form the World Council for Veterinary Health. So it's still very early days. We haven't really got a structure as such, but if there's anybody there who's watching, who James's normal audience, then if you are a vet, if you are concerned about what's going on, not just with these mod RNA jabs, but in veterinary medicine generally, or animal health or welfare generally, we will be trying to put together some sort of international body which will speak for the animals. So if anybody's interested in being a part of that, then please contact me. And that's really, and yeah, and if anybody wants to finance it, that'd be even better as well. So as I say, we're looking to establish the World Council for Veterinary Health to highlight these and other health issues in pets, livestock, but also sort of conservation and wildlife as well. So we're going to cover issues such as vaccination, feeding, neutering, intensive farming, overtraining, which is often happens too much too young in horses, overuse of chemicals such as parasiticides for flea treatments, and the harm that they're doing to the insects and the fish, etc. And also with the World Health Organization One Health policy, there are potential dangers to with mandatory procedures for animals. So we need to be there to be calling these dangers out. Obviously, climate change is being used to attack farming and the keeping of pets already. So we know that this is one angle that they're being used, that's being used to try and attack farming and replace livestock farming with solar farms and wind farms and whatever else. I mean, ultimately the aim is to reduce meat and dairy consumption down to zero and replace it with frankenfood. So, you know, we need to be aware of this. And if we really want to keep healthy food farming, then we've got to be on their case. I say it's already been stated incorrectly that all human infectious diseases come from animals. So we know this isn't true, obviously, but it's sort of laying the ground for future blaming and mandating and all the rest of it. So that's me. That's a very quick run through with background puppy noises for the risks that we're facing potentially from these mod RNA jabs if they are more widely rolled out for animals, especially from the food side of things, but also for our pets as well. That's my website there, my Substack, and my email. So if anybody wants to reach out to me, then please do.
That was fantastic, Roger. If you want to stop sharing, and I'll put that all in the notes and everything before we can close up our conversation with, you know, now is an opportunity for people to get involved in something that needs to be created. I mean, we need to have a focus worldwide. You know, you're the only vet I know that is thinking this way. I'm sure there must be others, but they are few and far between. One of the things I managed to do early on during COVID was just reach out to, you know, medical doctors who were treating patients better than they were getting, you know, from the standard operating procedure in the hospitals. And so, you know, just starting by reaching out to veterinarians who I'm going to venture to guess the vast majority know none of what you just covered, okay? To find vets who are open-minded. I can think of someone I know in Mexico. I can think of, I think you may know as well.
Yep.
So, you know, there are those around the world, but what we need to do is support the ones who are trying to treat animals more humanely, more reasonably, more, you know, healthfully. And, you know, anything that you can say to support what it is you're trying to do to spread the word, now is the time for people to do exactly that. So again, the floor is yours.
Yeah, thanks, James. You know, as I say, as far as I'm aware, I'm the only vet in the UK that's sort of been speaking out about it. I'm sure there are others who feel similarly, but you know, as we've seen with some of the doctors who spoke out, how they got treated as well. So I'm sure there's a fear there for people to speak out. But that's again the reason for us all coming together and creating a voice, where if we all stand together and we put out, you know, statements by the World Council for Veterinary Health as opposed to individuals, then, you know, there's less chance of retribution against those of us who think as we are. So, you know, I would encourage everybody to come forward. And, you know, I'm not just from the veterinary field, but also anybody who's involved with, you know, I've got where I get my raw milk from in the UK. John is the farmer there. He's going to help advise on farmer issues when they and farming issues when they come up for discussion. So, you know, if someone's, you know, heavily involved with horses and got an influential role, you know, obviously the more influential people that we can get on board, the stronger our voices and the more seriously we'll be taken. But you know, we do need voices for covering all aspects of animal health to try and and get this off the ground, and have eyes and ears, not just to speak out, but actually to have eyes and ears around the world, because very often things are going to be trialed. I mean, I know, for example, there's a mosquito trial going on in Hawaii at the moment, trying to spread Wolbachia bacteria in the name of conservation, but they haven't really checked it out
Mhm.
to know what's going on there. So, I would urge those people to, that's going in the courts at the moment, and it needs support as a venture as well. But obviously, if that was to be done, and then they could decide that actually other flies or other mosquitoes or whatever can be used to try as vectors to control other diseases, then we don't know what the hell they're going to try and release wherever. And yet pretty much every trial that I'm aware of where they've released cane toads or rabbits or wherever to try and as control pests, it's nearly always ended up with what they've released becoming far more of a pest than what they were trying to control. So, we've got a history of really screwing things up in the ecology, and you know so this is a wider issue than just the mod RNA and prions. Ultimately, that is an important one in the more immediate future, but there will be other issues that we think are going to be global problems as well that we need to get on top of.
You've touched on a lot of things. You can't simply cook your food and get rid of prions. That doesn't work. They're, much more resistant to heat. Even autoclaving, medical instruments doesn't make them go away. This is a very big problem. You mentioned that, these can even find their way into um, uh, vegetarian, food, plants, and so forth.
So,
Um,
yeah.
and so, thank you for all you do. I think you just put your little friend down. I was going to ask you if you could give him some screen time. And, this is the whole point. I mean, the ability to have, companionship and friendship with our, four-legged furry friends and whatever else you may have. Who's your friend and how long have you had him? And,
Yeah,
what kind?
this is Motley. So he's 12 weeks old. I've had him about just over 4 weeks now, and he is pretty well house trained because he was desperate to go out just before we started, and he let me know. So he really knew that he was supposed to be going outside to have a pee. So he's learning very quickly. Yes. So
It's these kinds of relationships, with our animal friends, are something that we have to put our foot down and say, no, you will not, harm the health of our loved ones, our family, and our extended family through pets and farm animals and so forth. So thank you for doing what you're doing. I appreciate it. And I'm going to do everything I can to spread the word. I'll put your information down there. People know how to get a hold of me. If anybody's interested, you can certainly call me at 310-619-3055. I'll get you in touch with Roger. We need to put together an organization of, veterinarians and people who, care for animals around the world, to head off this insanity before it takes hold even deeper than it is. So, Roger, thank you for what you're doing, and I
yeah, just
appreciate
one
it.
last quick thing I nearly forgot. I've actually had a paper published in the Journal for Independent Medicine on the use of PCR in
I'll
clinical
include that. You shared
observation.
that with me.
Yeah,
I couldn't agree more. The use of that is completely fraudulent. It's not a diagnostic, and your article is spot on. I'll make sure to include that for everybody down below. And
brilliant.
thank you for all that you're doing, and I hope we can help you do even more.
Well, thank you, and thank you, James, for everything you're doing too. It's always mutually appreciated, I'm sure.
Thank you.